Thank you, Dr. Ramanajan. So our next speaker is Dr. Ranit Aharonov. Dr. Aharonov comes from Pangea Biomed, where she serves as the Chief Technology Officer. Please help me welcome Dr. Aharonov to the stage.
Can you hear me? Okay, so thank you for the invitation. I think there's no dispute that precision oncology has really brought incredible advances in treating cancer patients. But the reality is that still the minority of patients benefit from precision oncology today. And our vision at Pangea Biomed is to democratize precision oncology and really make it relevant for all cancer patients. And we are doing this by shifting the focus from looking at single genes, at alterations of genetic coding at the level of single genes, to a much broader look at abnormalities of gene expression. And by doing that, we are able to nominate treatment to the vast majority of cancer patients instead of to the minority, and also get superior response rates. The platform we developed, which is called Enlight, can also improve drug lifecycle management and development at all stages. So starting from developing bespoke biomarkers for new drugs, through designing optimal drug combinations, all the way to label expansion and saving failed trials. As I'll show you, we have extensive validation of Enlight, and we're also starting a clinical trial, an interventional clinical trial at the NCI, sponsored by the NCI this year, which will study the clinical utility of Enlight. So before I tell you how Enlight works and the validation results of Enlight, I want to start with a true story. So this is a young patient, a woman, 35 years old. Around mid-2019, she was diagnosed with a very rare form of liver cancer called fibrolamellar carcinoma. Her disease progressed. She failed several lines of treatment. She was sent to an NGS lab for sequencing, and as often happens, the result was essentially report empty. So no actionable mutation, no actionable fusion, nothing that would suggest a good matching for precision oncology treatments. She continued to deteriorate, and then luckily she got to be treated at Professor Ra'anan Berger's at Sheba Medical Center in Israel, and he serves on our SAB. And he suggested that we should have an Enlight analysis of her RNA-seq data. And Enlight suggested that she's highly likely to respond both to anti-PD-1 and to anti-CTLA-4 drugs. And this was surprising because her standard biomarkers were negative. So she had no PD-L1 expression, low TMB, microsatellite stable. Nevertheless, given her condition, he decided to start her on ipi-nivo, a combination of anti-PD-1 and anti-CTLA-4, and she responded and went into complete remission. And today, more than 2 years later, she's alive and leading a normal life free of the disease. This was published very recently as a case report in the Journal of Immunotherapy for Cancer. And really our vision is to make this the norm rather than the exception, something that wouldn't merit publishing a case report on. So the question is, why is this the exception right now? The problem is that we're still looking, and we heard it here, somebody said we're preaching to the choir, so to speak, that today we're looking very narrowly at the drug target. So when we're considering whether a patient would respond to a drug, the focus is looking at the level of genetic aberrations at the drug target. But there's a lot more going on at the cell. And what we're doing is in Pangea is opening the aperture and looking not only at what is going on at the level of the drug target itself or targets, but rather at the entire context of the cell. So we are asking, are there other genes, you see the pointer, that functionally interact with the drug target, and by understanding the activity of those genes, we are able to predict whether the context in which the drug will operate for that specific patient, whether it is such that it will help the drug be effective or actually more inclined to develop resistance. So this idea of looking at networks of genetic interactions by looking at expression, the gene expression, is actually work that has been going on for more than a decade, started by our co-founders, Eitan Lupin. Today, Eitan is the head of the Cancer Data Science Lab at the NCI, Zev Ranit, and Eyal Gottlieb, who today is the VP of Research at MD Anderson. And I'll try in 2 minutes to tell you what the idea is, the basic idea, which Enlight is based on. So when we think about biomarkers, and people already discussed this in this session, usually what is done is that you take cohorts of patients that receive a drug, some respond, some do not respond, and then you use your favorite supervised algorithm and you look for what differentiates responders from non-responders. And the problem with this is that it's very specific. You have to do it again for every indication and every patient population, and data is very scarce. So there are usually very small amounts of data labeled with outcome for the drug, very hard to put your hands on such data. What is available is big data in cancer. There's a lot of in vitro data, there are a lot of knockdown experiments. There's a lot of human data. So there's a lot of expression data of tumors with overall survival without any outcome data for a specific drug. And the idea was that if we could identify, using that big data, pairs of genes that interact, so this is a very data-driven approach. We're not asking if those proteins actually interact in the cell. What we mean by interact is, for example, think of gene A and gene B. And you're searching for things like this. So if gene A is inhibited or underactive for any reason, it could be a drug inhibiting gene A or it could be a natural inhibition. And now if you find gene B, that when it is overexpressed, this actually together with the underexpression of gene A, this creates a decrease in the fitness of the tumor. This is an interesting interaction. There might be another gene B or C that when together with gene A, only when they're both together, that creates reduction in fitness of the tumor. So if we could find these pairs of genes, now if we had a drug inhibiting A, we could look at all the genes that interact in such a way with gene A and see the activity, overlay the activity pattern of the specific patient we're looking at, and ask whether the genes that should be overactive in order to decrease the fitness of the tumor are indeed overactive, and those that should be underactive are indeed underactive. And the way that we find these interactions is by statistical methods using this big data, which I don't have time to go into right now. But this enables us to calculate a matching score based on the RNA expression, gene expression of a specific tumor to any targeted drug. And notice that I never used anything that had to do with the data of the outcome of the drug. So I can generate a biomarker for a drug under development. I do not need to know anything about what happens when you administer the drug to the patient. All I need to know is what the drug is targeting. So Enlight, which is our platform, actually improves upon these earlier academic versions of the system and has been translated to a more clinical scenario. And just recently, our work is featured on the cover of the Med issue of January. Med is the flagship medical journal of the Cell Press. And what we show there really is validating Enlight on hundreds of cases. And in two interesting clinical scenarios. One is patient-centric, so sort of the same story I told you about this woman. There's a patient looking for a drug. The other interesting case is a drug looking for patients. So given a drug, what would be the best set of patients or the best subpopulation or the best indications to go after for this drug? So I'll show you just the main results from that paper. So what we did is we searched for clinical data sets that we haven't used before at all. So these are cases that were not used in the development of the idea in a more academic setting, nor when we developed Enlight and translated it into clinical scenarios. So all of these are cases from the literature, public cases. You can see different indications color-coded. You can see different drugs, targeted drugs, small molecules, immunotherapy, other monoclonal antibodies. And what we asked is a very simple question. In all these cases, patients received the drug that's written on the x-axis, and there was a response category given by the people who ran the experiment. And we set a single threshold on our Enlight matching score before we looked at this data. So all this data was, we blinded ourselves to the data, and we said if the score of a patient, say, receiving trastuzumab was above that threshold, we will call that treatment and light match to the patient. And we asked a simple question. Our patients, all these patients received the drugs written there, so they were already, in reality, getting a drug that was supposedly good for them, but obviously, as often is the case, many of them did not respond. Actually, the majority did not respond. Could we, using a light, stratify those patients to responders and non-responders? So are those patients that got the treatment and then light thought this is the right treatment for them, are they more likely to respond than patients who got the treatment and then light thought it is not the right treatment for them? And the way you measure that is by looking at the odds ratio of response between light matched cases and light non-matched cases. And this is what you see on the y-axis. And you can see that in essentially all data sets, the odds ratio is above 1, and overall we have an odds ratio of 2.59. The bottom just shows that not only is the PPV very high, the positive predictive value, but actually you can very efficiently exclude patients based on having a low light matching score. We also compared light to other biomarkers. So you see here many biomarkers, some of them are just prognostic, like cytolytic index, and light is actually better than other biomarkers. And for those immune checkpoint blockers data sets, the combination of light with interferon gamma is actually really strong. So this is something we're looking into for the future. But also very interestingly, for 4 of these data sets that we looked at, the people that, the researchers that published the paper developed an in-cohort supervised biomarker. So they actually did what I said at the beginning. They trained a classifier biomarker to differentiate between responders and non-responders. And supposedly this should be much stronger than a very broad, unsupervised approach as the one that we take, but in fact our results are comparable to that, which is really encouraging. So I want to finish up by telling you about our, so to speak, next generation of light. So the top here is what I've told you about before. So you take a tumor block, you do RNA sequencing or microarrays. We're pretty agnostic to the modality of measuring gene expression. You get gene expression, and then you do the light analysis and get an light matching score. But this is rather cost intensive and takes a lot of time, and we asked, could we skip that by trying to impute or predict gene expression from the image of an H&E slide? Which sounds a little crazy, but we were not the first to try it, and we collaborate with the NCI and the Australian National University on this. And instead of actually measuring RNA expression, we trained the deep neural network to take the image of the slide and predict or impute the gene expression. And then we hook up light to that. So light is agnostic to whether this is real RNA data or the RNA data that's imputed from the slide. And what we see is 2 things. First, if we look just at the correlation of the expression of the predicted expression by the deep neural network and the actual expression, we do very well and actually better than the state of the art today. There's one other group doing this, and we beat that state of the art. But more importantly to us, if you look at the bottom left, we found, we're still looking for more data to validate, but we found 6 data sets where we had the slide and we had the outcome. And again, we see that the odds ratio overall is above 2. So even directly from the H&E, using a deep neural network, if we use light, we get high predictive power, and it's even comparable to one of the supervised classifiers based on the actual RNA expression. So this is extremely exciting, and this could be a whole new way to really democratize precision oncology by skipping the RNA sequencing part. So just to summarize, we have basically 3 products in our pipeline. One is called a light lens. This is work we do with biopharma companies, helping them in life cycle management and drug development, as I said initially. We have a light match, which is what I told you about. This is already offered pro bono to patients that have RNA sequencing data, and this is what we're going to validate at the NCI. And Light DP, which is still in scientific and clinical validation, but this is our next generation of light directly from the H&E slide. So I want to thank you and thank the incredible team at Pangea. Do I have time for questions? One? Okay.
Maybe one short question, yes. Does anyone have a question?
Thank you.